Good evening, everyone. Welcome you all to the interesting session about transplant immunology. Is it the right match? As you know that in the current era where the number of transplant, especially the solid organ transplant, is increasing day by day, and there is an increasing awareness and requirement for especially the heart and lung transplant, especially for the end-stage lung disease and pulmonary hypertension. And as you know, for the transplant to be successful, there are 2-3 important things. One is the adequately matched donor, and second thing is that adequate immunosuppression, and third thing is the HLA matching. And to speak just a brief about what is HLA. So as you know, this MHC, or the histocompatibility complex, it's located on a small chromosome of chromosome 6. And there the small area is called the HLA. And it plays an important role in the different immune reaction. So how far we know the immune reactions and how far we are able to suppress, especially after organ transplantation, that plays a key role in the success for transplantation. Now, there are different HLA tests that we usually come across. That is one is HLA typing. Second thing is the HLA antibody testing, and then comes a cross matching. So everybody has a different opinion, and there is always a confusion about interpretation of this test. So I have with us the Dr. Manish Mourya. He's a PhD holder from AIIMS, and is a lab director of Histotech Research and Diagnostic Lab. So he'll be sharing us about different HLA tests and how to interpret the different HLA tests in the clinical context, and with a few case scenarios. So over to Dr. Manish sir for the presentations.
Thank you, Dr. Vimi. And it's very glad to have this opportunity. I just wanted to share my screen. Yeah, my screen is visible now.
Yes, sir. Yes, sir. Your desktop view is visible, sir.
Yeah. So, yes. Yeah. So currently I am an associate consultant with the Newberg Group also, and lab director for Histotech Lab. So today topic is very interesting. I think in a lot of ISOT meetings and the midterm meeting, we had a lot of discussion, but still this paradox and paradigm has always been there, how we look at the patient once it's sensitized patient, and how we discriminate between the sensitized and non-sensitized patient, as well as post-transplant monitoring. So for that, we need to understand the compatibility between the patient and the donor, and basically the histocompatibility. Histo means the tissue compatibility. So as we understand, the adaptive immune system develop under the pressure of human leukocyte antigen. This is very important slogan, where the sentence saying the human body and HLA system has a capability to tolerate other organs or develop antigen, means tolerance against the antigen, as well as the reactive quality. So how this HLA work? HLA has a different segment, and with this segment, HLA recognize peptide as an antigen, and this peptide come across the immune repertoire system, and immune repertoire system further give instructions to develop the antibody mediated response or the T cell mediated or cellular mediated response. So there is a two kind of a response: cell mediated response, antibody mediated response. We are going to talk about the antibody mediated response, because this response is more involved in rejecting the organ, very fast reacting, and once the antibody come into a picture, it's very difficult to save. Cellular immune response still can be managed with the drug, but antibody mediated response is little bit very tough for managing. So when we understand about the HLA system, HLA is a class 1, class 2, and class 3. There are 3 kind of a system which present on chromosome 6 is a small arm, 21.31 region. So very small region has a huge long chain, almost 6 million nucleotide is there, which is going to synthesize one molecule as a protein molecule, which is going to recognize more than 6,000 or maybe higher n number of antigen and generating the response against those antigen to clear out those antigen, to clear out, to understand those antigen, to develop the tolerance against those antigen and to reject those antigens. So tolerance and the rejection, there is a 2 things we have to manage to the tolerance and the response to create a graph or a camera in any transplant, any kind of a transplant, even a bone marrow, even a solid organ, lung, tissue, any kind of a transplant. So here we are observing one picture is class 1 made with the alpha 1, alpha 2, alpha 3 domain and the beta 2 domain. Alpha 1 and alpha 2 is responsible to interact with antigen. The same way class 2, alpha 1 and beta 1 is responsible to interact with antigen. Class 2 has a wide range of antigen interaction because the groove is open. So long chain of epitope can go and bind with the class 2, whereas the class 1 is a evolutionary conserved. It's developed during the course of evolution, during the course of a life for any restricted antigen binding. Now we have to understand there are certain terminology. So what is a codon? Codon is a 3 nucleotide sequence which can translate mRNA and protein. What is an exonic region? Exonic region is a protein coding region or a functional region of a gene. Each exon has an intronic region. Intronic region is sometimes very long region. So that region is split out or alternate split out to decide the control of a production of a protein. Means it's controlling the production of a translation. So this is how the protein comes into a picture with the central dogma. DNA to mRNA, mRNA to translation as a protein. Now we can look at there are certain terminology which frequently written into a report like a locus. So locus is a specific region where we are deciding the multiple form of antigen present. Allele is a alternate form of a gene. Like if long is one character, then how long of a particular person is decided by a long gene. The same way, if HLA has a one antigen like A2, so how much variation A2 antigen like A204, A206, A211, A224, whatever. So how these antigen has a grading and capacity to enhance the immune system or tolerate the immune system against any antigen. Genotype means everyone is having a 2 chromosome. Each chromosome has a 2 gene. So combination of a 2 gene is called genotype. Phenotype means the expressional characteristics of the genotype. So if we look at the 4-digit data, I will come on that topic. Okay. And the polymorphism, we know how gene is polymorphic. Homozygous means both genes are same. Heterozygous means one gene is different. One gene is different on the both chromosome. Yes, haplotype. So there are a certain word is coming like haploidentical match, identical match. So haplotype is a set of a gene which came from the one parent to the offspring. So like if father has a set of a gene and B set of a gene, mother has a C set of a gene and D set of a gene. So sibling can be A, C, A, D or B, C, B, D. So this kind of a distribution is called the haplotypic distribution, and this is very strictly followed in HLA system. Very rarely the cross reactivity happen, cross reaction happen. And this, we have observed very few cases where the cross matching happen, cross matching happen, and gene jump from one parents to the other parents and making some different haplotype. So on the basis of a basic genetic knowledge, the nomenclature has decided, and nomenclature come in this way for an HLA locus. So suppose if we have a HLA-A locus is representing the gene, or loci, both terminology can be used for A locus. Star is representing the typing done through the molecular method. If a star is not there, that means the typing done by the serology method, like with the Terasaki plates. Then the first digit, this is second field, this is third field, this is fourth field. So entire HLA nomenclature is on the four field nomenclature. The first two field is defining the allele group. The second field is called 4-digit. Nowadays, second field, very frequently scientific community is communicating between each other, but 4-digit data or of 2-field data is very common to address this nomenclature. So this is second field data where HLA specific protein are coming into picture. So this both two is defining the exonic variation based protein formation and translation. Now the third field is a synonymous variation in exonic region. Synonymous variation means the variation within the codon is not going to make any difference of making the protein. Only configuration changes is observed. So sometimes 6-digit data may value, but there is no parameter where we can address that 6-digit data is really influencing and involved in producing the antibody, or we can monitor that 6-digit data can be influencing the understanding of antibody and antigen interaction. But definitely the configuration is changed, and this is completely research oriented. And fourth field is defining the non-coding region, means like intronic region, and also defining the nature of expression of a HLA molecule. Like N means null allele, molecule is not going to express over the surface. If somewhere like an L means low expression, H means high expression. So there are a lot of way to express the expression of the HLA molecule over the surface of a cell. So now we know that the HLA nomenclature and HLA antigen. Now we need to understand whether patient and donor, how we are going to select those compatible donor and the patient. For that, we need to monitor antibody. Antibody monitoring is in 3 basic ways. First is a CDC cross-match, second is a flow cytometry cross-match, and third is a Luminex DSA. Luminex DSA, lysate-based DSA. So right now I'm going to talk about the donor cell involvement and serum. In CDC cross-match, what we do, we take a serum, recipient serum, we take a donor lymphocyte, and we add on a complement, and we look at the complement mediated cell death, observe with the dye, where if the dye enter in influence of complement and addressing the pore over the cell, it's look like dead cell, it's look like a dead cell, or it's a bright cell. So this is very basic CDC cross match each and every laboratory is doing to understand, but this is very less sensitive, lot of time and negative report coming. Now this next level of sensitivity is a flow cytometer. So in flow cytometer, again, we take a donor cell and the sera. We incubate with the donor cell and the sera, then add a secondary dye, IgG fluorescent dye, and add the identification of a different kind of a cell like CD3 marker, CD marker. For entire lymphocyte, CD3 marker. For T cell, CD4 marker. For B cell, CD19, CD20 marker. So in that case, we can observe those markers along with the IgG antibody interaction and giving a and looking at the channel shift, where 50 channel shift this is observed above the 50 is in class 1 because the dye fluorescent is lower in compared to B cell, and in B cell the channel shift is more than 150 observed. But there are a lot of flyers, need a very high technical staff to interpret the report and result. Most of the time, class 2 comes as a false positive. Most of the time, reason is because B cell can interact with non-specific antibody. Also, some drug effect like rituximab. So this kind of a scenario where the B cell comes positive. Lot of the lab is using CD19. CD19 always comes positive when the patient has a rituximab. And so for that, their alternate CD marker is a CD20 and CD22. So we can ask for the CD20 or CD22 to look at, and they can predict whether it's a specific HLA antibody or non-specific antibody. So flow cytometer is an interaction with the dye. Now comes to the Luminex. Luminex, where we use the bead, but we are using a live cell also. So we prepared the lysate, burst the cell, converted cell into an antigen. We drop antigen or incubate antigen with the bead, and bead is going to bind with the antigen. Then after we incubate with the sera. So same in CDC and flow, we are taking a live cell. In Luminex lysate also, we are taking a live cell. Only the thing is we are converting live cell antigen and transferring on the bead, a specific kind of a bead, which are going to give a specific result about the HLA. So in Luminex DSA, we are going to get a specificity about the class 1 and class 2. No non-HLA, no other antibody we can observe. So this way with the Luminex DSA, we can avoid false positive result in compared to CDC and flow cross match. But CDC and flow cross match can give an overall understanding of an antibody, whether the patient's serum has any kind of antibody which are going to interact with the donor. So that's why always recommend to go for at least 2 methods of a cross match to understand the antibody and antigen compatibility between the patient and the donor. Right. In nowadays, there are a lot of laboratories available everywhere. So good to go for all 3 kind of tests before the pre-transplant, so that if any technical error or anything is there, it cannot be missed out. Okay. So now we know that the flow cytometry is a T cell is channel shift is about 50 and the B cell channel shift is about more than 120. This is how the flow cytometry result we interpret. Now we are now developing another flow cytometry method. I think it is going to be available in next couple of a month, within a 2-3 month, where we can club the Luminix DSA formula and the flow cytometry formula. So we are going to work on a bead as well as cell together. So we get to know that the overall non-HLA antibody, autoantibody, along with the specific HLA antibody class 1, ABC, class 2, DQ, DP. So this is very extensive test which is under development. Within 2-3 months, it is going to available into the market. Laboratory is starting. This is how we are interpreting the DSA report. So DSA class 1 and class 2. The raw MFI value is class 1 is 170 and the class 2 149. Lysate control is this, positive and negative lysate control. So this result is negative. In this second, the MFI raw value is more than 2000. So this is considered as a positive in compared to class 1 has a very low. When we look at this report, this is also very high, class 1, 5,000 something, and class 2 is low. So this is how we are getting the data, converting those data into the reporting format and giving as an MFI positive or negative in DSA, lysate-based DSA. So this is cell-based antibody monitoring, CDC, flow, and Luminix cross-match. Now cross-matches, this cell-based cross-match part is over. We are going to have a virtual cross-match. So how virtual cross-match works? So in virtual cross-match, again, the instrument involvement is a Luminix only. There is a 3 kind of a virtual cross-match available. First is a screening virtual cross-match, very cost-effective, low informative. Second is a specific cross-match, but intermediate level, like low resolution, 2-digit data, 1-field data. We can overall understand that what antigen and antibody, what kind of antibody present in the patient and in class 1 and class 2. And the third one is a specific as a single antigen every time we are talking about it. So in screening, there are one bead has a multiple antigen. Number of bead is low. So we just predict out that there is some antibody present and which can interact with the certain number of antigen. So in that case, lot of time it's showing positive class 1 and class 2, but it doesn't mean that it is donor specific. For donor specific, we have to go for ultimately single antigen. PRA is also giving idea about 2-digit. So we can predict something about with PRA, but single antigen is a complete conclusive test. Lot of lab is not doing intermediate PRA. Reason is the cost is almost same. Lab is charging 20,000 nearby the PRA and 30,000 nearby the single antigen. But at one go, it can be clear out. In solid organ transplant, specifically lung and heart transplant, we are recommending both the PRA and the single antigen because this can give a clear picture and the clinician will not suffer at the time of a cadaver transplant if any other lab is saying this is positive, this is negative. So both way, we are confirming that patient doesn't have an antibody or patient has an antibody and data is matching with each other. Okay. PRA screening and PRA low resolution is little less sensitive. Lot of time, if single antigen is coming positive, not MFI is so high, then PRA screening and PRA low resolution can possibly come as negative. So clinicians are understanding why it is because one is coming as negative and single antigen is showing is positive. So MFI has to look at MFI, if 2000 to 4000 is varying, PRA can come positive. So this is how the structure of a bead, class 1 and class 2. PRA screening, multiple color of a bead, multiple antigen has coated. And PRA low resolution, number of a set of an antigen, hardly 3 to 4 antigen is coated. And single antigen is only one kind of antigen. In our PRA screening, we are giving MICA positive and negative also. So because we are using a certain kit which can give a class 1, HLA class 1, class 2, and MICA understanding also. In screening, and cost is also very efficient. Means it's less than 5,000-6,000, like that. So this is how. This is a low resolution PRA data. So here we are looking at the low resolution like B24, B44, A66. So if suppose a donor has an antigen B44 and patient has an antibody against B44, then we have to look at the high resolution 4-digit data. Whether the donor B44 is B44O2 and the patient has a B44O2 or some other antibody or not. But if suppose donor has B44 antigen, but patient doesn't have a B44 antibody at all, then we not need to look at the single antigen data. Because patient doesn't have at low level itself antibody. Low resolution antibody is not present as similar as patient data, donor data. The same reporting with all kind of positive, negative stats, biostats we are giving from the laboratory. And this is class 2 data. Now this comes to the single antigen. So in single antigen, this is how the screen board of analysis look like, where the positive control, the negative control, the positive control, negative control ratio, this all are very important because this is going to give an idea about the clinicians and the laboratory where the data is authentic or not. How the interpretation has done, how the tail analysis has done, how the epitope analysis is done. So this all analysis we are doing in our laboratory before releasing any data. So this is how the class 1 and this is class 2. Now we need to understand the cross-reactive and epitope management. So how epitope and cross-reactive is different. When we talk about the cross-reactivity, that means antibody present in the recipient can interact a multiple antigen in the donor. When we talk about the epitope reactivity, means antibody present in the recipient can interact with the multiple donor antigen or one antigen different epitope also. So when the kit, when this entire system has designed, then they choose the different epitope of a particular antigen. They form those epitope over the bead as an antigen, and those epitope is going to interact with the antibody of a patient. So epitopic understanding with the patient antibody understanding is going to analyzed by the software, and then software is giving an idea about the MFI value, the antigenic property of serum in single antigen or any antigen monitoring in any antigen monitor. So if we look at the epitope variation from one antigen to the another antigen coated on a bead, then we can figure out, yes, this is matching between the two antigen, what we are giving as a DSA positive, or both are different epitope in a different antigen, which we are giving a DSA positive. This is all interpretation is done through the software. Now the second thing is a cross-reactivity. Cross-reactivity is defined during the evolution of understanding of antigen-antibody interaction in HLA nomenclature system. Actually, this is the one of the thing which initiate the scientists which working on a HLA antibody understanding from beginning 1901, almost 120 year. This all research has started before 121 year. This research has started, and the first they found that, okay, the patient serum has something which is going to interact with the donor. Then they rule out patient serum has antibody, some antibody, which are interacting with the donor, and that antibody is in a group of different antibody form, which is interacting with the group of a certain population. Those population has a certain different kind of a protein molecule. It's called antigen. Now they started differentiating that each antibody is interacting with the multiple antigen. So they are segregated class 1, class 2, and class 3 in DR, class 1 A and B, as a cross-reactive group. Here, if we look at this very common example, like A locus 28. If donor has 68 HLA antigen and patient has 69 antibody, the 68 and 69 fall into a same cross-reactive group. That's why the 68 antibody can interact with 69 donor antigen. So in single antigen in PRA, we are looking that number is not matching, but still the reaction is there. Still the CDC is coming positive or the flow is coming positive. What could be the reason? So this is very common reason where the number is not matching. But if we look at this chart is showing that this is coming into a same cross-reactive segment. Okay. Now, in same way, one example we can take here in B locus, where if the donor has 38 and patient has 39 antibody, they are interacting with, the patient sera is interacting with the donor antigen. There are certain fade lines also, there is a group also. These all are referring to the different strength of a cross-reactivity, like the strong line is strong cross-reactive, a faded line or the small line, less thick line is a cross-reactive, and this entire group belongs to the cross-reactive group. So we look at this line and this green line. The entire group, we are looking in very rare situation where the CDC and flow is coming positive. Otherwise, we lose a potential donor. So that's why we are not looking this entire box. We are started looking from the green to thin line and then in the box. The same way, the DR also. Now, if we look at this picture, then the understanding about is, if the sensitivity of a virtual cross-match started from 1000 MFI and it goes to more than 10,000 to 14,000. Here, the single antigen is lying. 10,000 to 1,000 to 2,000 above, the single antigen started as reflecting positive antibody. against the donor or DSA positive. This 2000 to 8000 is a flow positive. Flow positive can come in this region. Mostly I observe that if 4000 below the MFI or CDC or DSA single antigen is coming, MFI single antigen MFI is around 4000, mostly flow is on borderline or negative. 5000, 6000 is showing borderline, just exact, and especially in the class 2. Class 1 is still it can come around 4000 as positive, but class 2 the range is from 4000 to 6000, and class 1 is range from 6000 to 8000, 5000, 6000 to 8000, where the class 1 can come little earlier, 4 to 6000, flow cross match start coming positive. And the CDC came positive in this region. More than 6, 7,000, 8,000. And above this is also started coming negative, because in that case, the prozone effect come into a picture. So different dilution has to perform for an CDC cross match, and also flow cross match is recommended for go for a different dilution. Lot of lab is not doing the different dilution because it's not cost effective. So again, they have to discuss with the clinician, and clinician has to and then go for an at least 1:4, 1:8, or 1:10 level of dilution in flow cross match. And CDC, most of the lab is doing once it's required. So if flow and CDC is negative, is still antibody can possible. To rule out this, we need a high sensitive test, and the virtual cross match is a high sensitive test along with Luminex lysate based DSA. This is very interesting article, 8,000 almost heart transplant recipient they have observed, and where they have categorized into a different PRA percentage, and they observe that the odds ratio and the confidence interval is high in 0% and decreasing above the 25%. The survival and the rejection within the first year. So if any patient has a 25 more than 25% can able to develop antibody against the donor, because the system is active, the certain antibody, supporting antibody can present, which cannot be reflected in pre-transplant because of maybe MF, the binding capacity of antibody is low, or the titer of antibody is low. So this can be enhanced at the post-transplant. So they have observed this kind of a risk factor above the 25%. But this data is mostly suitable for the high homogeneous population, where the antigen and antibody circulation within the population is similar antigen circulation within the population is high. In India, India has a very diverse antigen population. Chances is very low to match one person to other person, even 0.001%. So for a cadaver transplant, if we look at this data, this kind of a data can tell us how we are conscious for the post-transplant monitoring. Side by side, we have to not lose the potential donor. So these two things we have to keep in mind of looking the PRA percentage in the report. So this is the timeline where the post-transplant monitoring can perform. This is related to solid organ I have made for the renal transplant. Most of the people are using this all. But after post-transplant, a new antibody is developing, then CD4, CD8 is coming, CD marker is coming into a picture. CD means like complement system, CD4, C4D, C1D, C1Q. So this all come into a picture and enhance the antibody mediated rejections. This is how the post-transplant memory work. If the patient has a low post-transplant antibody, it can enhance pre-transplant antibody. It can enhance the post-transplant antibody because there is an MFI, there is an antibody, but the titer is low in pre-transplant, can increase in post-transplant. So this is how it is. And I'm not going to cover this non-HLA right now because non-HLA is not in practice and a lot of lab is developing those protocols. So this is how. And this test also we are doing for an CYP antibody for a drug metabolism, understanding of a drug metabolism for tacrolimus. Thank you. Hello. 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 Hello.
Sir, I think there is a technical glitch from the Varghese sir. He will be joining in 2 minutes.
Okay.
Hi, Dr. Manish, are you able to hear me? Hi.
Yeah.
Hi. So while Vimi starts to join us, I'd like to ask you a few doubts.
Okay.
So when we see SAB reports, the reports we have here in India, they say an MF5 value cutoff of 1,000. Now, but when we go on to international articles, we see their MF5 cutoffs are like 2,000. So is there a standard cutoff value world over, or does it like vary from lab to lab?
So MFI value for a lower cutoff is not varying from lab to lab. This is well set as within 1,000, below the 2,000. Below the 2,000.
Yeah, so anything above 1,000 would be positive, and
anything above the 2,000
would be strongly positive.
So yes, so 1,000 is positive because as an understanding of a post-transplant memory, but not restricted to the transplant.
Okay, so now the second one would be about the lower resolution HLAs and the higher resolution HLAs.
Okay.
So supposing we have a donor and we have a recipient, we get around 6 to 8 hours before we are actually in the OT to harvest the lung. So when we do a low resolution HLA and do a virtual cross match, if we get a positive cross match, virtual cross match, we actually don't have time to actually go and do a higher resolution one. So that's a practical problem we face as urgent. So we need to clearly know if a virtual cross match is positive, because sometimes we may not do the donor at all. So would you recommend doing a lower resolution HLA and then going on to a higher resolution, or do a higher resolution every time?
So
because,
honestly,
it takes
I'm
around
telling
6
patient.
hours.
Yeah,
Because I feel it takes around 6 hours for the report.
right. So I understand this problem because a lot of time when the cadaver come into night or that lab is facing the challenge. But when we look at the data is a 4-digit data is only the comparable data for the antibody point of view. Right? Right.
Yeah.
4-digit data, if any facility has a Luminex, they can predict out. Not required for an NGS. Right. So Luminex based facility can is a sufficient for the cadaver transplant understanding till the single antigen. Only the interpretation who is interpreting the low Luminex data, their interpretation should be confident to interpret the 4-digit data. So because Luminex is a low resolution platform, but can predict out the 4-digit data. Chances of error in 4-digit data in Luminex is not more than 5%. Right. So any lab who is doing in a
No, sir, you continue, sir. I wanted to ask after this point. No problem. Continue, sir.
Yeah. So now the question is whether we go for HLA typing for a donor, high resolution data, not required for a cadaver if we have a 4-digit HLA typing data. Considering 5% can vary. First question. Second question. If we have a low resolution 2-digit data and patient has an antibody is exactly matching to the low digit 2 data, only then need to understand the 4-digit data. Otherwise, it's not required to understand because cross-reactive chart to the direct donor understanding can be predicted out with the 4-digit data, at least 60% cases. Unrelated 60% cases.
Sir, but when we usually come across this, especially the low resolution HLA, most of the time what we have is the 2-digit pathway for most of our reports. So, so in that scenario, sir, because the ideal what you're saying is a 4-digit 4-digit resolution HLA, which is it possible in low resolution or is it possible only in high resolution, sir?
So low resolution has the 2 methods now the laboratory are using. One is the Luminex based HLA typing. Second is a SSP. In SSP, you cannot predict 4-digit data. But in Luminex, you can predict the 4-digit data. One understanding. Second understanding, suppose patient has antibody against A24. Okay. But donor has antigen of A11. Then we don't need to look at the donor 4-digit data because at the 2-digit data, it's ruled out that patient doesn't have a donor specific antibody. Clear?
Yes, yes, sir, yes.
So like first we have to rule out patient has antibody at 2-digit. If yes, then we have to go for a 4-digit to understand, to rule out exactly antigen antibody DSA is there or not.
Okay, sir. Other few things what doubt what everybody has is especially regarding the different PRA, sir. I mean, not about the PRA, especially about the Craig and the splits. So when we see the report, sir, like when we see the report from, I mean, from a user point of view, how do you understand it is a Craig and about the split, sir? Because from your diagram, I think, I mean, we are able to understand little more in detail about the cross-reacting, pathway and the splits. But when we get the user report, how to identify it? Because I'm a little confused about that.
So I'm just going to show, because this kind of a report only we are generating from our laboratory. No one is, 1 minute. Screen share I have to, and then. Yes. So in our report, whenever we are saying that cross-reactive, this graph you will get every time. When the donor information is available, then we are releasing this as an additional page.
Okay.
So in that we address this and this. This is only this our laboratory is doing. None of the laboratory is releasing this page.
Okay, sir. Sir, in that report, that same report, sir, if you can take the same report just one more time, I think which you already shown.
1 minute, yes. Yes.
No, sir. No, sir. The diagram, sir. The diagram.
Okay. Yes.
Yes, sir. So in that report, you can see that there is a true positive, false positive, false negative. How to interpret that? That different
So
true positive, false positive.
Yeah, true positive, false negative comes in a low resolution PRA format. Okay. Always, not in single antigen. Single antigen only it came as a tail analysis, right? Positive with the MFI value. Reason is in PRA low resolution, the bead has a multiple antigen, and there is a combination of a bead is there. So software and antibody is going to bind on a bead. So we cannot differentiate over the bead which antigen it's binding unless and until we have a different combination. So like A1, A3, B4, B1, B2, one bead, A1, A2, B2, B4, second bead. Now the software is going to interpret like if both is positive, both bead is positive, that means A1 is positive, right? But if B1 is positive and second bead doesn't have a B1, that means A1 cannot be positive, B1 is positive. So differential combination based interpretation by the software is giving. And now what is the tail analysis? Because bead has a different antigen and we are writing those antigen in one single line. So it's called line antigen, and we are doing an analysis. So its line is look like a tail. Within the tail, which one is positive?
Okay. Okay.
So like PRA, if I show you the PRA report, this is single antigen. Yes. So if you look at this, this is single antigen. 1 minute. So this is how the bead structure for the PRA. This is how the bead is structured. So one bead has a multiple antigen. Out of that, which one is a positive is analyzed by the tail analysis. So like this bead has a 29, this is also has a 29. Okay. And this is not having a 29. So if these two are positive, likely chances to 29 has positive. But if these two are the positive, then this doesn't have a 26. So the chances of 26 either positive or something else is positive. So this kind of a combination in PRA, that's why the tail analysis is recommended to go for.
So to I understand more, sir, like now if you take the B60 class 1 specific antigen, the true positive is 3. That means at 3 times it is truly positive among the beads, sir, or how do you interpret that value 3, sir?
Yes. So true positive 3 means the 3 bead is coming positive.
Okay.
3 place B60 was quoted and the 3 point is coming positive. B7, 1 place is quoted and 1 place is coming positive. Okay. So that's why if we look at this, so MFI is also varying. If 3 is positive, MFI is high. If 1 is varying, MFI is low. Because antibody titer is low. So number of the bead showing presence of a particular antigen and particular bead in how many number has acquired.
Yes.
So one bead has almost 5 to 10,000 in number.
Yes.
And one antigen has a multiple on bead.
Okay.
Like that.
Sir, one other thing is that what most of them has a doubt is, I think is regarding the disparity when the PRA is almost nil and multiple antigen positivity in a single antigen bead. How do you feel how we should interpret it in that clinical context, or how to rectify reading that report, sir?
So PRA, this is completely based on MFI understanding. PRA, if the single antigen has a low MFI value, like if any lab is reporting single antigen A24 MFI around 3,000, 4,000, okay, or let's say 2,000, 2,500, PRA can be negative. Because the sensitivity of a single antigen to interact with the antibody is very high in compared to PRA. PRA needs a more titer of antibody comparing to single antigen. Because in PRA, there is a competition also to binding with the antigen. So this is, bead is acting as similar as a cell is acting into the immune system. As similar, not exactly as similar.
And sir, when the MFI, it's if it is coming to more than 7,000 or 8,000, in that clinical context, when the PRA becomes nil, how do we go ahead, sir?
This is not possible.
Not possible.
Not possible. If any laboratory is giving this, then we have to request them to rerun the sample, or we need, they have to treat the sample, or sometime the titer is like if any laboratory is giving a PRA, a single antigen positive around more than 10,000 or 15,000, and PRA is negative. that means titer is very high and they have to do the dilution based assay to prove that PR is also coming positive because of the prozone effect.
We'll ask our audience, sir, because I think time is getting near. We'll ask our audience whether they have any questions, 1 or 2 questions.
Yes, sir.
I think there is no question, sir. So finally, sir, I wanted to ask your comments, sir, about especially in a center where we don't have a very good immunology lab, the way to go ahead in the cross-matching by different methods. What is your final comment? Comment regarding the different cross-matching methods when one is positive and other is negative, or let's say the flow is positive and CDC is negative, and with a virtual cross-match, if it is taking a lot of time, how do you comment about going ahead, sir?
So very simple, very simple. CDC can positive, can negative, and so many times. There is a lot of up and down where the lab is reporting CDC negative. At the time of cadaver, the technician is very much in pressure because they are doing overtime in a lot of lab, especially for the night. Okay, so they can do the mistake. I'm not denying it. Most of the lab has a control system, but it still can possible. So at least 2 method of cross match is important. Now, CDC, let's say whatever the result come, either positive or negative. If positive, we can't go ahead without ruling out why it's coming positive. It can be a autoantibody, it can be a non-specific antibody, need to rule out. How we are going to rule out CDC is positive and that positivity is a true positive, or CDC is negative, we need to know any positive with the other high sensitive test because CDC can be a false positive. So we have 2 things. One is a flow cross match, one is a Luminex based DSA. Flow cross match, most of the time B cell is coming as borderline positive. Lot of the lab is reporting false B cell positive. So many times you have observed that B cell is positive, T cell is negative, and DSA and CDC is coming negative. Right. So that borderline positive, we don't need to worry about it. We have to just go for a DSA, Luminex based DSA, because this borderline can possible with the other antibody like bacterial antibody, viral antibody. If B cell can interact, B antigen can interact, because HLA class 2 is responsible for the exogenous antigen. So it can interact, the same antigen can mimic as a HLA antigen and interact with the class 2. Can possible. So we have to go for a DSA Luminex based. Luminex based DSA rule out any non-specificity, only giving a class 1 and class 2 HLA antibody and this.
Okay, sir.
So if CDC and DSA is available, that is more than sufficient for go for a transplant, for a cadaver point. But there is a certain discussion was going on because a lot of the lab doesn't have a good quality control on the Luminex DSA. Luminex DSA is almost 60% manual procedure. CDC and flow is 100% manual procedure. So 100% means everything is designed by the lab. Protocol is designed by the lab. Reagent control is not there. They are buying few reagents from the one vendor, from the other vendor, like that. Okay. So this is all 100% combination. Their lab is making their own kit and producing the result. DSA has a control. It's internal control also, internal lysate control, positive control. So more reliable in terms of understanding of HLA antibody. But DSA is not going to give non-HLA understanding. debate is whether we are really interested to look at non-HLA antibody during the cadaver transplant or not. According to me, we should not look at, unless and until patient has any antibody disease, any autoimmune antibody or any very high PRA. If high PRA is there, if MICA is positive, or non-HLA antibody is positive in pre-transplant, then only flow is, if coming a positive, is very conclusive. So that we are not doing in practice, I think so. No one is doing. Non-HLA we are not looking at all. So CDC and Luminix DSA is more than sufficient. And
okay,
HLA typing of the donor.
And just
comparing with the single antibody.
Yes, sir. Finally, before ending the discussion, sir, because we also had previously one non-DSA AMR. So do we do the non-DSA typing, sir, to know the type of what do you call, type of like KU or NU or all those things? Do we do the typing?
Post-transplant, look at the non-HLA DSA is very good practice. We have to look at actually every 6 months, but this is additional cost for the patient. And it's more costlier than the single antigen. So that's why Indian laboratory is not able to keep their diagnostic setup, because kit is very costly. Expiry date is very low, 3 to 4 months is expiry date. So if number of the clinicians started recommending, then it's very worthful. So once society has decided, okay, we are going for a heart transplant or lung transplant, post-transplant after this time, or at least once in a year, we have to monitor the non-HLA. Then laboratory can keep, and it's feasible for the laboratory. So this kind of amendment, if can come as a general consideration, then can possible to add those things.
Okay, sir. Thank you for the nice discussion, sir. I think we should have frequently a discussion, especially in this immunology, so that a physician and the surgeons in the transplant team will be more aware about how to interpret the report with the clinical context in mind. It was a wonderful discussion, especially in many aspects. We were, especially regarding the cross match, there was a lot of confusion, and thank you for clearing many of our doubts. I think there are no more questions in the chat box. Yes. So, so thank you, Dr. Manish, for sparing 1 hour of our time. And thank you everyone for the listening, and hope we had a nice interactive discussion. Thank you all.
Thank you, Dr. Vimi. Thank you, Dr. Balasubramoniam.